All right, good morning, everybody. It is a little after 10, so I'm going to go ahead and get started. Again, thanks for everyone for being here online this morning, and I apologize for the last minute change of plans. But we'll continue with our coverage of the brain today in a kind of normal lecture format. Today's class was actually not too difficult to transition to an online format. So there is no class program for today, since you had your exam on Friday, but we will pick up and continue with the brain. And here's what our agenda looks like. I feel like we say this all the time. What are we going to do today in class? And the answer is the same thing we try to do every day, Pinky. I don't know if anyone's ever watched Pinky and the Brain. You can appreciate that one, but obviously the main character is the brain, which is appropriate today as well. This is what our trying to take over the world looks like today. I've got a couple announcements for you about some scholarship and prize opportunities that may be of interest. We're going to do a quick review activity with drawing, not surprising, and then we'll continue with the brain lecture. And I will wrap things up sharing the overall exam scores for exam number 2 that you took on Friday. You know your own score already. And we'll wrap things up with a ticket out. So let's go ahead and I've got a couple announcements for you. First, there's 2 prizes offered by the biology department for graduating seniors. The first is for anyone interested in going to graduate school or professional school. And there is it's a $500 prize, basically $500 cash that you can use towards graduate school or whatever, really. The QR code is here. If you have any questions, let me know. You can see the prize winners from the past 2 years on the slide here. Additionally, there is an opportunity to help you pay for a field course. If any of you are not graduating seniors and thinking about doing some kind of field course or research experience this summer, there is a Rice Award that funds, actually, I don't know what the funding limit is for this, but it's a decent amount of money. If you're thinking about doing field work, go ahead and scan that QR code or let me know, and I can pass that information along to you. All right, so we've got a review activity here. Before I start into it, let me just pause and see if you have any questions. In the online lecture format, the best way to ask a question is just to go ahead and unmute yourself and speak up. You can also use like the virtual hand thingy, but it risks I might miss that. Just go ahead and speak up. So before we go into it, what questions do you have? All right, well, here we've got a 2-minute drawing review activity. As best you can from memory, draw a diagram of the brain from the lateral aspect and label the cerebrum, the diencephalon, the cerebellum, the brainstem, and the spinal cord. Now, I know we've only so far covered the brainstem in any detail, but we did look at an overall diagram of the structure of the brain and specifically the 4 major structures of the brain. So please take 2 minutes and go ahead and draw that as best you can. Once you've got that drawing down, you can go ahead and consult your notes, make sure it looks good, make any tweaks that you'd like to make. All right, next 2 minutes, quick review drawing for you. You should have produced something that looks like this. Remember that the cerebrum surrounds these other structures. So you can see if you look carefully on this figure, which is taken from your textbook, you can see how it's made to look, the left hemisphere of the cerebrum is made to look transparent here. So that you can see the diencephalon here in this bluish purple, the post-rostral part of the brainstem, so the midbrain and the part of the pons here in teal, and then part of the cerebellum here kind of in orange in the back. We are gonna continue walking through these structures today in class. Last class we covered the brain stem. Today we're going to move to the cerebellum. All right, so let's do it. Here is the cerebellum. Here is a sagittally transected image of the brain, and you can see the brain stem here, you can see the cerebrum here, and you can see the cerebellum is basically this giant cauliflower looking thing on the back here. It's about a tenth, or I think it's around 11 or 12% of the mass of the brain overall. And it is just dorsal and posterior to the pons and medulla. So it's basically just behind those two topmost structures, excuse me, the two bottommost structures of your brainstem. And to aid you along as we look at the cerebellum and the diencephalon, I've made you another study guide. You can go ahead and use the QR code here. And actually what I think would probably be easier is if I put the link in the chat for you. So let me do that. Sorry, give me 1 second and I'll make sure that's in the chat. It's probably easier to get than it is through here. There we go. I'll put a link there, and like the other guides I've made for you, it's a series of Google slides you can download and save as your own. Some of the labels are blank so that you can fill it in, but it will help you kind of keep your notes organized and hopefully be useful to you as we work through this material. So go ahead and get that guide going. I'll give you a second to grab it. I just posted the link in the chat as well. And we're going to talk about the cerebellum. Here is a view of a posterior view of a cerebellum. So this is from if you're standing behind someone looking at the back of their head. And you can see why I said it looks kind of like cauliflower. Actually, it looks a little bit like noodles or something like that. Note, importantly, I'm going to start with the major divisions of the cerebellum. There are two cerebellar hemispheres. And as you might guess, they're the left and the right hemisphere. And they're separated by something called the vermis, which is a structure right down the middle. You can't really see it too well from this perspective, but I'm going to show you a sagittally cut cerebellum in a second, and you can see it better. Now, each of the two hemispheres is then broken down into three lobes. There is the anterior lobe, kind of up here, the posterior lobe in the back. These two lobes are primarily involved with coordinating trunk and limb movements. So these are the areas that's integrating motor output from the cerebrum and sensory input from the rest of the body to coordinate your movements through space. The third lobe is called the flocculonodular lobe, which is fun to say. The flocculonodular lobe, you can't actually see in this image. It's ventral to the posterior lobe, so it's basically kind of where my cursor is here, but in front of the structure. And it is important for maintaining your overall equilibrium and also helps coordinate your head and eye movements. So again, the cerebellum is divided into two cerebellar hemispheres, the left and right, and each hemisphere has three lobes. The anterior and posterior lobe you can see here, as well as the flocculonodular lobe, which is kind of internal. Actually, let me change the diagram here so you can see that. Here is the same, actually you can't see the flocculonodular lobe here, but I'll show it to you in a diagram. I think it's on the next slide. This is the same view of the cerebellum. So this is a posterior view, but now we have a coronal section of it. So it's a posterior view sliced down the coronal plane. And you can see here that when we do that, we have three kind of distinct tissue regions inside the cerebellum. And you can see the spinal cord kind of emerging down here. Remember, the spinal cord is continuous with the medulla oblongata, which is just anterior to this part of the cerebellum here. Internally, each of these lobes has three regions. Oh, excuse me. The first is the cortex. The cortex is this outer layer of gray matter. So again, remember, your central nervous system, gray matter is primarily interneurons, short unmyelinated interneurons, the cell bodies of your motor, in this case, well, yeah, often, at least in the spinal cord, the cell bodies of your motor neurons, lots of cell bodies here in the central nervous, in the brain as well. And also, of course, as with any white or gray matter, lots of neuroglia. So this cortex is the outer layer of gray matter, and this integrates all the information coming into the cerebellum. We then have what's called the Arbor of Vitae. And you can see it's this kind of white branch-like structure going through the middle of the cerebellum. It literally translates as the tree of life because it has these tree or branching-like structures. This is white because it contains the axons of neurons transmitting information to and from the cerebrum. So those axons, just to refresh your memory, are myelinated, and that myelin sheath that surrounds those axons to help insulate it, to help keep those electrical signals moving efficiently and quickly, that myelin sheath is white. And so that is why this arbor vitae, this branching tissue you see here, is white. And finally, we have what's called the deep cerebellar nuclei. So remember the term nucleus singular, nuclei plural in this context refers to areas of gray matter where we have groups of often of the cell bodies of different neurons. And so this is gray matter that is situated deeply, so that as opposed to the superficial gray matter of the cortex. And in this case, these nuclei are important relays for, again, from information from the cerebellar cortex to other parts of the brain. Again, most often coordinating the movement through space, your movement through space. Now let's take a look at the cerebellum from a different direction. This is a parasagittal section. So I promised you earlier, I'd show you the floccular nodular lobe, and here you can see it. And you can see how it's situated. So just to make sure we're oriented, this is a parasagittal section through the cerebellum. So that's along the sagittal plane, but not in the middle. And we're looking at this from a left lateral view. And so here we have the midbrain that you're familiar with, the brainstem that you're familiar with. You can see the midbrain, the pons, the medulla down here. We walked through that last week. Cerebellum is just posterior, and we have the three lobes of the cerebellum. The anterior lobe, you can see colored in blue up here. It's the superior and anterior most region. Then we have the posterior lobe, which is the majority of the cerebellum. It's orange here, and it is the inferior and posterior region. And then we have this little guy right here, kind of situated just between this posterior lobe and the ventricle here, and that is the flocculonodular lobe. Now, importantly, remember this left ventricle right here. We talked about it in the context of the pons. It's closely associated with your brainstem, and it's in fact between the brainstem and the cerebellum. And remember, these ventricles are filled with cerebrospinal fluid, so they're, again, to make some connections, they're lined with those ependymal cells. That is the epithelial cells of the neuroglia. They have the little cilia that keep the, I like making this hand gesture for cilia, that keep the cerebrospinal fluid moving. And here, note, we have this choroid plexus. We'll see several different plexuses throughout the brain associated with ventricles. And these are sites where that cerebrospinal fluid is made via a process of filtration from the blood. We're not going to get into the details of how that's done, but I do want you to know that the choroid plexus is this structure where the cerebral spinal fluid is produced. And so, take us a step back, parasagittal section, focusing on the cerebellum, anterior, posterior, and flocculonodular lobes. And you can see here those structure inside the cerebellum. You can see this arbor vitae, this kind of branching white material. And then on the outside, we have this gray matter in the form of the cerebral cortex. Let me pause here and see what questions you have before I talk a little more about the cerebellum. Okay, I mentioned earlier that the cerebellum's primary function, but not its only, is to process information to smooth out movement. So this is all about hand-eye coordination and something called motor memory. So those of you, especially if you're a musician or an athlete in some way, this part of your brain is really important because this is the part of the brain that learns these repetitive motor tasks so you can do them really well every time. So something like throwing a fastball or playing the violin or whatever specific skill that you are employing in sports or music or other fields, of course, but those are two that come to mind. The cerebellum is where all of those very fine scale movements and sometimes not fine scale movements become coordinated. And so this slide is a little word heavy. I'm not crazy, but I don't feel like you need to copy down every word. I'm going to just walk you through some of the general structures and their functions here. And this information is also in your textbook. First, I want to talk about the motor cortex of the cerebrum. So this is a part of the brain we haven't talked about yet, but part of the cerebrum that controls motor output, sends information through the pontine nuclei in the pons. So we talked about those nuclei in the pons involved in relaying information from the cerebrum to the cerebellum. Here they are. So the motor cortex from the cerebrum sends information through these pontine nuclei and then through the middle cerebellar peduncles. We talked about the peduncles, we talked about the brainstem. And that information then gets sent to the lateral, anterior, and posterior lobes of the cerebellum. Furthermore, information about your head position and your equilibrium, and that information comes from your inner ear, that travels through the vestibular nuclei in the medulla. So again, we talked about some of these specific nuclei in the brainstem. So this is one example in the medulla. As well as information from proprioceptors throughout your body. So muscles, tendons, and joints, that comes to your spinal cord, and that travels information both from the vestibular nuclei in the medulla, and this information from your spinal cord travels through the inferior cerebellar peduncles to the flacconodular lobe, and then on to parts of the anterior and posterior lobes. You don't need to memorize all of these pathways. I want you to just have a general understanding of what information is transmitted to the cerebellum and how it gets there. I want to back up for a second and make sure, actually, I think I'm going to go forward here and make sure, as I'm talking about these peduncles, I want to make sure that you can see them. Oh, here we go. I'm going to go back to this diagram. I want to make sure that you can visualize the superior, middle, and inferior cerebellar peduncles. We talked about these a little bit in the context of the brainstem, but now we're talking about them explicitly because this is the center of, this is where information is transmitted from these nuclei in the brainstem to the cerebellum. And specifically, we have information from the motor cortex of the cerebrum that travels through the middle cerebellar peduncles, so right here. And then we have information from both the spinal cord as well as from the medulla, or excuse me, from the your ear, travel through the medulla, through the inferior cerebellar peduncles here. Information goes to the flacconodular lobe right there. And then finally, we have the cerebellum sends information out back through the superior cerebellar peduncles to the motor cortex of the cerebrum. So in other words, there's constant crosstalk between the cerebellum and the motor cortex because the cerebellum is integrating all of the sensory information about where your limbs are in space, and it's sending that information back to the motor cortex to help you coordinate your movements. And that information travels through the superior cerebellar peduncles, which are right here. You can see in you can see in this diagram right here. But the takeaway message from this slide, what I want you to know is that information is transmitted to and from the cerebellum through these middle, inferior, and superior cerebellar peduncles, and that information travels then to the cerebrum, onto the brainstem, or down through the spinal cord. One important thing to note is all of these fibers going to and from the cerebellum are ipsilateral. So this is a vocabulary term we learned at the very beginning of class, the first week, but we haven't used too much throughout the semester. And all it means simply is that information from the right side of the body is processed on the right, in the right hemisphere of the cerebellum, and information on the left side of the body is processed on the left side of the cerebellum. Now, this is contrary to information from the cerebrum, which is contralateral, meaning that information to and from the left side of your body goes to the right hemisphere of your cerebrum and vice versa. So the cerebrum is contralateral. The cerebellum that we're talking about here is ipsilateral. In addition to coordinating these muscle movements through space, I should say the movements of your body via muscle contraction through space, the cerebellum is also has some involvement in some higher cognitive functions. So I guess closely linked to that is this idea of muscle memory I mentioned earlier, so these muscle skills. And also, interestingly, some functions involved with cognition in language, problem solving, and task planning occur here. And there's centers elsewhere in the brain where we'll see where executive function, your ability to kind of think consciously, plan out actions, solve problems, and things like that happen. But some of that actually happens in the cerebellum as well. So as I said, cerebellum primarily involved with coordinating your movement through space, but also has a role in some of these higher functions such as motor skills and cognition and planning. I'm going to pause here on the cerebellum. That's about all we're going to cover with the cerebellum. But as a little bit of a whirlwind there, let me see what questions you have before we move on and talk about the diencephalon. Okay. Good job, everyone. I'm going to go back to this big diagram. So this is a diagram I presented in class last week. And recall, this shows you the major regions of the brain and the developmental regions from which they originate and then what they become in adult structures. So we'll often hear, just to walk us through this list again, a little bit of review, telencephalon becomes the cerebrum, that's this part of the brain here. The diencephalon developmentally then becomes three structures, the thalamus, the hypothalamus, and the epithalamus. That's what we're going to talk about next. Something called the mesencephalon becomes the midbrain, and the metencephalon becomes your pons and your cerebellum, and your myelencephalon becomes your medulla. Now, what's a little bit confusing about this is that in the adult brain, three of these structures that are derived from three different developmental regions, we all group together as the brainstem. So the midbrain, the pons, and the medulla become our brainstem. The cerebellum, even though it's derived from the same region developmentally as the pons, we call its own thing here. So just refresh your memory about these terms and how they're related. And note that this structure, the diencephalon developmentally, we still call it the diencephalon in adult brain, and specifically it's composed of these three structures. So let's take a closer look at your diencephalon. The diencephalon is nested kind of right within what I would consider kind of the center of your brain between your two cerebral hemispheres. And it is a central core of gray matter that is surrounded by the central hemisphere. So the diencephalon is this primarily, actually, is it exclusively gray matter? It might be even exclusively gray matter. There's very little white matter, if any. And it is composed of three main parts, something called the thalamus, something called the epithalamus, and something called the hypothalamus. Now, this makes it a little easier to remember because if a structure has thalamus in it, you know it's part of the diencephalon. And also, these terms give us a little bit of clue as to where these structures are located anatomically. The thalamus is kind of this big structure here in the middle. Remember, this is all gray matter. The epithalamus, epi meaning on, is this little bit right back here. I guess it's not really on top of, but it's kind of sticking out of. And then the hypo meaning lower or under, that's this part right here. So let's take a look at this. Let's take a step back and see what the structure looks like. So this is your brainstem here. You might remember this. We've got the medulla oblongata, your pons, and your midbrain here. And sitting right on top of your midbrain, nestled between your two cerebral hemispheres that you can't see, is your diencephalon. And that includes the thalamus that you see here, and then the hypothalamus. Can't really see the epithalamus. It's just back here behind this optic tract. And then we have two other structures that I won't put into too much detail right now, But we do have something called the infundibulum, which is this little stalk right here, and then the pituitary gland. A quick aside about the pituitary gland, it is a gland that is very important. I was going to say the most important, maybe even your most important endocrine gland. Now, we haven't talked about the endocrine system in detail in class yet, but this is, of course, where hormones are produced. And the pituitary gland is where lots of hormones are produced and stored, and it's very directly regulated by the hypothalamus. Specifically, hormones travel down this little stalk, this infundibulum, to the pituitary, and the pituitary releases all kinds of hormones. I forget exactly how many hormones in the pituitary, but somewhere around a half a dozen or more hormones are either stored here and then secreted or produced in the pituitary. That's a little aside. One other connection I want to make is, remember, the pituitary gland sits right here in this little cup that's called the sella turcica, which is part of your sphenoid bone. So as you recall from lab, I kept pointing out the structure whenever we looked at a sphenoid bone. I said, hey, that's the sella turcica. That's where your pituitary gland sits. And here's seeing that from the other side of the pituitary. Let's walk through the structures of the diencephalon. We're going to start with the thalamus. Most of your diencephalon is the thalamus, in fact, about 80% of it. And what's important to note is that you do have a left and a right thalamus. It's often referred to as just the thalamus, but there are a right and a left section, and they're united by this, what's called the interthalamic adhesion. And that is this kind of structure right here that bridges them. Sometimes that's called the intermediate mass. And so a left and a right thalamus with this intermediate mass. So you can surmise that this is left and this is right. So this is a posterior view of your thalamus. This is the brain stem here. You can see the midbrain here, and you can see your pons structure. And here's that choroid plexus associated with the fourth ventricle. And here's your left and right thalamus sitting on top of that. Now, the thalamus is composed of about 12 nuclei, and each of these nuclei has axons that extend to different parts of your cerebral cortex. This diagram is in your book. We do not need to know all of these nuclei. What I want you to know about the diencephalon is that it's composed of these nuclei, and these nuclei are divided by white matter. I mentioned earlier that there wasn't much, if any, white matter in the diencephalon. There is some white matter, and they kind of divide these nuclei into these different groups. So these are the myelinated axons that go to and from these nuclei. We don't need to know the names of all of these or anything like that. What I want you to know is the overall structure, that the thalamus contains all of these nuclei, and each of these nuclei is in close contact and communication with the cerebral cortex. What's important is generally the information that's relayed through these nuclei is not only sensory information going to the the cerebral cortex, but all input from any other part of the brain is relayed to the cerebral cortex through these nuclei. So we consider the thalamus is often considered the gateway to the cerebrum. So any information coming from your midbrain or for your cerebellum has to travel through this, the thalamus, this structure of the diencephalon, to get to the cerebrum. So it's considered the gateway because it doesn't send all of that information up to your cerebrum. So you're constantly, the part of the thalamus's job that I think is actually kind of fascinating. Its job is to filter out information and only deliver to your cerebrum what is important in a given moment. So this is why if you're in a noisy room, you can focus on one person speaking and ignore everything else. This is also why you don't consciously think about lots of things that are happening with your body all the time. So for example, until I say this, none of you are probably thinking about the way your shirt feels on your skin or your clothes or the way your body feels sitting in your chair or anything like that, because the diencephalon, and specifically the thalamus, is not delivering that information to your cerebrum because it's not important in the moment. But now I've just drawn your attention to it. Things have changed. Now your diencephalon is delivering that information to your cerebrum so you can think about it. So it's a really important information filtration center. Let's see here. I want to talk a little bit about let's continue talking about the hypothalamus. If we look at the external anatomy of the hypothalamus, let me see here. Sorry, let me pause for a second. Yeah, let me transition and talk about the hypothalamus. So this finishes up the thalamus. Let's move into the hypothalamus, but let me pause and see what questions you have before we do that. And actually, let's do this. I'm looking at the time. I'm going to skip ahead just a little bit here because I'm kind of covering a lot. And let's do like a quick 1 minute stretch break. Go ahead, take 1 minute. We'll be back in action. I encourage you just to stand up from your chair, unfocus your eyes on a computer screen for a second, give your brain a rest, and we'll get back in just a second. All right, you've got a good little stretch there. Appreciate that little break. I know I just stretched out a second. Let's get back to the hypothalamus. Let me see here. So actually, I want to move back and forth between a couple diagrams here. The hypothalamus, remember, is this structure that is kind of superior and rostral from the thalamus. So let me go back here, show you what that looks like in this diagram. So remember, this is a lateral view. We have the hypothalamus is this kind of region right here. And if we look at that from another angle, we'll see that region right here. So this is kind of an inferior view. So this is kind of looking up your brainstem here. Here's your pons, kind of at the underside of the thalamus, and your hypothalamus here. The pituitary gland is cut off in this figure. And there's a couple important structures that we can see here very readily in brain dissections that you'll see tomorrow in lab. And that's your optic nerves that branch off from here. They're of course cut off here. And something called the optic chiasma, where the information from the eyes crosses over to go to your cerebral hemispheres. Don't worry about those structures too much right now. I just want to orient you to where the hypothalamus is anatomically and how we can see it from an external view of the brain. Now, just posterior to the hypothalamus are these two round bodies called the mammillary bodies as well. There's where the hypothalamus is located. Hypothalamus, like the thalamus, contains all of these different nuclei. These are centers of gray matter that are associated with different functions, both different sensory and motor functions. And you don't need to know all of these, just as I'm not asking you to know them from the thalamus, but I want to show you this diagram so you get an idea of what this thing looks like. Remember, these are color-coded to show you the nuclei. They don't really look like these jelly beans inside your brain. A couple of things that are important to take away from this is that we do have this mammillary body, as I said, that's just posterior to your hypothalamus. It's kind of between the hypothalamus and the brainstem. We'll see this mammillary body on the sheep brains that we're going to look at tomorrow in lab. We also see this, the anterior, what's called the anterior commissure is like this tissue that connects the two sides of the hypothalamus. We have the optic chiasma, that's where these optic nerves cross over, and I showed you that from a different view before. And then the pituitary gland kind of hanging down here from this infundibulum. And all of these nuclei in here, I'm not going to walk through those in detail. If you take a look at a whole brain from the inferior view, you can see this stuff quite nicely. Our spinal cord going up into our medulla oblongata. You can see the pons here. And as we continue moving anteriorly, you can see these two round nubs, the mammillary body. Then you can see the optic chiasma here, and where the optic nerves cross over. So these are major landmarks that you'll see in our brain dissections tomorrow. And our hypothalamus is kind of tucked right in here between these mammillary bodies and this optic chiasm. These nuclei in the hypothalamus play a variety of important roles, and I'm just going to give you an overview of them here. The hypothalamus is important for regulating your body's functions around things that you don't think about. So it controls a lot of the autonomic nervous system, or it's part of the autonomic nervous system, a better way to say that, in controlling such functions as your heart rate, your blood pressure, your salivary glands, importantly also included in your emotional responses. So these are things that, you know, I guess in my mind, that's a little bit of an in-between, like, is your emotional response something you consciously control or not? Yes, you kind of can, but it's integrated in feelings of pleasure, rage, fear, for example. It regulates your body temperature, so controlling whether you're sweating or shivering. Also, your different drives, so it's important for your hunger and thirst, your body drive to eat or to drink, of course. Also involved with your sex drive. It regulates your sleep-wake cycles, and the hypothalamus is kind of the master control center for your endocrine system, which of course regulates all of these other important autonomic functions throughout your body. You know, I got ahead of myself a little bit here. This bullet point up here is hunger and thirst sensation. So you're integrating those feelings of hunger and thirst, but then at the same time, it regulates the motivational behavior related to that for eating, drinking, as well as your sex drive. The nuclei associated with the mammillary bodies are important for memory formation as well. So the takeaway message here is the nuclei and the hypothalamus are importantly regulating autonomic functions that you don't think about, as well as your emotional response and things like your behavioral drives to eat, drink, and sex. Now, final structure of the diencephalon is the epithalamus. The epithalamus, I said that it's not really located on the top of the thalamus. It actually, it's called the epithalamus because it sits on top of the third ventricle, one of those ventricles filled with the cerebrospinal fluid. And it's smaller, and it is, I don't want to say, it's associated with this choroid plexus as well. So this is another location where this cerebrospinal fluid is made. So the epithalamus is associated with this choroid plexus, but also importantly includes something called the pineal gland. And you've probably heard of the pineal gland before. The pineal gland is regulated by the hypothalamus, and it secretes the hormone melatonin. Now, melatonin is super important for maintaining your circadian rhythm, so your sleep-wake cycle, and also plays a regulatory role in terms of your sex drive. So that interacts with the hypothalamus to affect that. Melatonin is also often thought of kind of generally as your sleep hormone. It's the hormone that's released at the end of the day, kind of regulates when you get sleepy. Now, we could talk about that in a lot more detail, but I will just say briefly is that there's different people have different circadian rhythms related to melatonin. That's why some of you are what is called like night owls. You like to stay up late and you feel like your brain is at its most capable, maybe late at night. Maybe you like staying up really late and then sleeping in a bit in the morning. Other folks tend to get sleepy earlier in the day and then wake up earlier, and their brain is kind of at its peak functioning capacity earlier in the day. What's important to note too is this changes throughout your lifetime. So little kids, for example, tend to fall asleep or get sleepy earlier in the evening as a general rule, and then get up earlier in the morning. Anyone that's been around a lot of little kids know that they like to get up a lot earlier than you do, especially on Saturdays and Sundays. And at the same time, when you're around your teenage years, that shifts pretty dramatically, and it's harder for you to fall asleep in the evening. You're not sleepy till pretty late in the night. And that becomes problematic when, say, for example, high schools start classes super early in the morning. So a lot of teenagers are stuck in this cycle where their body is basically telling them they're not tired until pretty late in the evening, but then every morning they still have to get up really early anyway. I know I slept for at least the third of my time in high school. I spent sleeping at my desk, which probably wasn't the best. Well, there's some other life decisions associated with that, but I'm going to blame the melatonin produced by my pineal gland, which remember is part of your epithalamus, which in turn is part of your diencephalon. That's my story about melatonin, and that about covers all we're going to say with the diencephalon here. So whirlwind tour of the diencephalon and a whirlwind tour of the cerebellum. I'm going to pause and see what questions you have, and then maybe talk a little bit about the cerebrum. Okay, well, let's see, I have a ticket out. Let's talk a little bit about the cerebrum. I'm going to give you an introduction with this, and we will continue at Wednesday. So your cerebrum is also known as your telencephalon. Notice that you're developmentally, you have this region called the telencephalon that develops in the cerebrum. So we kind of use these terms interchangeably. Generally, when we're referring to the structure in an adult human, we call it the cerebrum. I'll mostly call it the cerebrum. Far, this is the largest part of your brain. If someone told you to close your eyes and picture a brain, this is probably what you would picture. I'm sure you're not going to picture the epithalamus. It's the bulk of your brain, right? And what's important is that it's broken into, I shouldn't say broken, that's the wrong term. We separate it into two hemispheres, a left and a right hemisphere, that is divided by something called the longitudinal fissure. So there's deep division between the tissues of the left and the right hemisphere. We also have something called the transverse fissure, which is right here, that divides your cerebellum back here from your cerebrum up here. So the takeaway message from this slide is that the cerebrum is big. It's divided into two hemispheres by the longitudinal fissure here, and it's divided from the cerebellum by the transverse fissure here. Now let's take a closer look at the cerebrum. We're going to spend a little more time on the cerebrum than other parts of the brain because it's bigger and more complicated and does a bit more, right? What's important to note is, generally speaking, when we look at these hemispheres, they're not smooth. They have these grooves and ridges, right? And what that does is it serves to increase the amount of gray matter. So we have this cerebral cortex, which is this layer of gray matter that you see here. And it follows the contours of the external surface of the brain. So if there are lots of grooves and bumps in that external contour, it increases the amount of this gray matter, right? And so kind of the bumpier, the more grooves there are and the more bumps there are, the more gray matter there's going to be along that surface. And we use two important vocabulary terms to describe this topography. One is called a sulcus, or plural sulci, which are these grooves. And then we refer to a gyrus or gyri, plural, which is these ridges between the sulci. So sulci are the valleys, gyri are the hills, right? And what this does, as I said, is serves to increase the amount of gray matter. And there are some important sulci and gyri that we're going to talk about in a little bit. But I'm going to leave that as a cliffhanger. I'm going to leave it there for today. You're not going to know what those important sulci or gyri are until Wednesday. Of course, they're in the textbook. You can find them out if you're really dying for knowledge. But this way, you'll come to class and you'll be super excited because you'll be like, oh, I finally get to learn these important gyri and sulci. But I'm not going to tell you today. Instead, I'm going to give you an overview of how the class did on their exam, which was on Friday. It was open book. Basically, you guys crushed it. I was kind of incredulous when I saw the scores. Here's a histogram. As always, the range of scores is on the x-axis and the number of people that scored a certain value on the y-axis. So 10 or so of you got somewhere between a 97.5 and 100% and so on. The class average was a 96 out of 100. So yeah, I made that exam way too easy. You're welcome. Great job, everybody. Well, maybe I made it easy, or maybe you all just did an amazing job. Either way, I hope it went well. It go well for the vast majority of you. If you're finding your scores in this lower end here, know that even though that's a respectable score, depending on your own standards, that I just want you to be aware of where you fall out with the rest of the class, and know that this exam being open book and multiple choice, it is definitely going to be on the easier side compared to upcoming assessments, for example, your lab practical. So with that, I want to wrap things up. I have a ticket out for you. I have an image of the brain reminder again and again and again that our final exam period is on Friday, April 29th, and we'll have our lab practical then. I'll give you details as we get closer, but make sure you reserve this date and time. I will be back on campus. I'm going to actually leave campus now, and I will have open hours today between noon and 2 if you want to stop by. Otherwise, go ahead and upload your image from the beginning of class and see if you can answer a couple questions without consulting your notes. And I'm actually going to post this link. I think it might be easier in the Zoom world here to just post this in the chat. Whoops, sorry. See if you can answer those questions without consulting your notes. It's kind of a test for you and for me. Again, thank you everyone for your flexibility and for logging in online this morning. I will post a video for this lecture shortly. I'm going to stop recording here, but I will stay online for a few minutes to answer any questions you have. One last reminder, don't forget you have a lab quiz tomorrow. It is worth only 4 points, but it will be a closed book lab quiz in a format that you're familiar with. Basically, I'm going to put a skull bone in front of you and I'm going to have you tell me what it is. So make sure you're prepared for that in lab tomorrow. Otherwise, I will see you during open hours this afternoon, or I'll see you tomorrow in lab. Thanks again, everyone. Have an enjoyable rest of your day.